Hey everyone, thanks for joining me today for episode 785 of People Behind the Science. I'm your host, Dr. Marie McNeely, and I'm excited to welcome our guest, Dr. Jonathan or Jony Kipnis today. This episode is made possible with support from our sponsor, Innovative Research. Listeners, Innovative Research has provided scientists with quality lab products made in Novi, Michigan for more than 20 years. Their portfolio includes human and animal biologicals, assay and ELISA kits, antibodies, and more. Products can be purchased directly from their website by credit card or purchase order at www.innov-research.com. That's www.innov-research.com. They offer small quantities, bulk purchases, and custom orders. From human whole blood to specialized antibodies, Innovative Research has what you need to succeed in the lab. And today, Jony is going to tell us some great stories about his lab and his life. So get ready to meet another one of our brilliant people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am excited to be speaking with our guest scientist, Dr. Jonath Kipnis. So, Jony, welcome to the show today. How are you?
Thank you very much. Thanks for having me. I'm doing great.
Well, thank you so much for joining us. And before we get into the details of your work, I'd like to start by telling our listeners a little bit about you and how you got to where you are today. So listeners, Jony is a BJC Investigator and the Alan A. and Edith L. Wolff Distinguished Professor of Pathology and Immunology. He is also a Professor of Neurology, Neuroscience, and Neurosurgery, and Director of the Brain Immunology and Glia Center at Washington University School of Medicine in St. Louis. He received his bachelor's degree in biology from Tel Aviv University and his master's degree in neurobiology and PhD in neuroimmunology from the Weizmann Institute of Science in Israel. Jony worked at the University of Virginia for over a decade before joining the faculty at Washington University in St. Louis, and he has received numerous awards and honors, including an NIA MERIT Award, the NIH Director's Pioneer Award, the Harrison Foundation Distinguished Teaching Professorship in Neuroscience, a Gutenberg Research College Fellowship from the Johannes Gutenberg University of Mainz, and the Distinguished Research Career Development Award from the University of Virginia. In addition, he is a member of the National Academy of Medicine. And Jony, today we want to get to know you as a scientist, of course, but also as a person. So can you tell us, what do you like to do when you're not doing science?
I think when I'm not doing science in the lab or on computer, which actually usually when I do science, it's not really in the lab anymore, unfortunately. I do it on my computer or talking with scientists in the lab who do science anyway. But to answer your question, I love to read a good book and I love to have a good glass of wine and I love to cook and then to eat. So grilling is my passion outside of science.
I like it. And you mentioned reading as well. We love talking about books on the show. Do you have a favorite book that you'd like to recommend for all of us, whether it's related to science or not?
It's very hard to define a favorite book, right? Because there are so many books that I like. But since we are on a science podcast and since we are talking about people behind science and talking to hopefully scientists or future scientists, budding scientists, and I happen to be at home in my desk, I always have next to me a book by Edith L. She is a Nobel laureate, a neuroscientist Nobel laureate who escaped from Nazis in Germany, came to the US, actually came to St. Louis and was professor here at WashU for many, many years. And she got her Nobel Prize for discovering neurotrophic growth factor, NGF, which is a very important molecule for neuronal growth, and etc. So she wrote a wonderful book. It's called In Praise of Imperfection, My Life in Science. It's very well written. It's really beautiful. Unfortunately, it's discontinuous, so you cannot really buy a new book. You can only buy a secondhand. They're quite pricey, but libraries usually have it. We have one in our library in the lab. I have my private one at home. It's just an incredible book, super inspiring, and just the way it's written, it's so well written. So I highly recommend. Again, it's not to answer the question of what's my favorite book, but this is a book that I would definitely recommend to people, and also because I see it in front of me.
Amazing. And believe it or not, you were actually the first person to recommend this book on our show. So I will have to add this to our list for our listeners to find there, In Praise of Imperfection by Rita Levi-Montalcini. And now we mentioned that you are oftentimes doing science, particularly at the computer these days. But let's talk about your science. How do you describe your work to people who are outside of your field or perhaps outside of science altogether?
So we are neuroimmunologists. And basically, we are working on the intersection of the immune system and the nervous system. Actually, it's a funny story. I was undergraduate student in Tel Aviv University quite some time ago, and I was working in the biochemistry lab. And this professor tried to retain me in the lab for my graduate work. So he told me that biochemistry is the place to be. You will be in immunology or in neuroscience. Nobody knows anything what's going on there. It's such a new field. It's all just be developing. Biochemistry is solid. Mm-hmm. And I said, wait a second. Okay, so immunology and neuroscience. Okay, note taken. So I know where I want to be. And then when I joined the Weizmann Institute in Israel for my graduate studies, I happened to listen to this professor, Michal Schwartz, and she was describing some crazy ideas about interaction of immune cells and brain cells. And even though I really dreamt, since I remember myself, to cure cancer, I decided to give it a try and try her lab. And I fell in love with the field, and I never really left the topic of neuroimmunology. Back then, in the 90s, it was not a very popular field to be in. It was actually a quite unpopular field to be in. But today, it is blooming, and everybody realizes and understands that the brain and the immune system are not false. They interact constantly. Brain controls immune system activity. Immune system affects the brain function. They are mutually working together to ensure our organism's survival and well-being. And I don't think anybody is asking anymore whether or not two systems interact. I think the question is now is how do they interact? So I think it's a really exciting time to be in neuroimmunology. But again, I was coming from those dark times when neuroimmunology was not a fun topic to study.
Certainly. I think now, absolutely, you're right there. Neuroimmunology is this budding, growing, dynamic field to be in. And I'd love to talk about motivation. You spoke previously about this book that you keep by your side by Rita Levi-Montalcini. I think she's certainly an inspirational figure in science. Are there other people that you look up to yourself, or people who perhaps helped you get to where you are today that you look to as role models or mentors?
You know, I have a bunch of role models. It's funny, I call them godfathers and godmothers of science for me because I was never really trained with them, but they've been always there and they were supporting me throughout the years. And I don't know if I want to name them. I mean, they know who they are. I don't want to name them so that, maybe if I miss somebody, people will get upset. But I'll tell you what, I derive a lot of inspiration from people who are working in my lab as well. Some of these, I call them kids, they're not so kids, I mean, they're in the 20s and 30s, but some of these people are just absolutely incredible individuals. First of all, they're brilliant, they're super creative. Some of them, or actually many of them, come from middle-class working families. And actually, I was just talking with one guy, and he was telling me how he grew up, and played soccer without shoes because family didn't have money to buy him shoes, that kind of level. And then they reach all the way to the top of the science. He went to Hopkins for his PhD and then came to WashU for his postdoc that he's doing with a collaborator of ours and also working with us. And it's just incredible. You talk with these people and they tell you their life stories and they come with these crazy ideas that I myself wouldn't think about. And then they implement them in the lab and they show that yes or not correct. And then they are reshaping those ideas and we're discussing a lot. And it's just absolutely incredible. So I do get a lot of inspiration from these people as well. So I don't think it always needs to be somebody who is a super high achiever. I think sometimes you just get inspired by people who are on their trajectory. Even sometimes an undergrad, I would talk with an undergrad and I was like, wow, oh my God, this is incredible. He is so smart, brilliant, and so creative and just thinks in different dimensions. And so these things are very inspiring.
Well, I love that you're looking for and finding inspiration in those around you. And I think these people can be tremendous sources of motivation. And for me, at least, it's helpful to have a little quote or a saying or something someone said to me in my mind as I go through a difficult time or even just to make it through the day sometimes. So do you have a mantra or a quote or a saying that really keeps you motivated?
I have a bunch, actually. So first of all, I have worked for many years at the University of Virginia. And of course, there, everybody's obsessed with Thomas Jefferson, as was I, absolutely obsessed with the man. And well, we know all founding fathers. But I think Jefferson had, of course, a very special place in my heart for many reasons. And so to this day, I keep, of course, a bust of Jefferson on my desk, not in the office. You may think I'm crazy. And also, most importantly, his book of quotes, his Bible, there is Jefferson Bible. which is actually Morals of Jesus Christ Without the Miracles, which is quite incredible. A book to read, very, very thin, but very exciting. But I also keep a book of quotes of Jefferson, and some quotes there are extremely inspirational. And once in a while, I just go through and I'll just read a couple of pages from there. But I do love quotes. So I have on my door in the office, I have a quote from, yes, you guessed correct, Edith L, which says that without intuition, there would be no science at all. And I agree with her 100%. I think that intuition is a very important driver in science. And I know that a lot of things that I do and we do in the lab are driven by intuition, my intuition, intuition of people around me. Also, I love there was a quote of Carl Sagan, which said that something somewhere is waiting to be found, he said, and I think we're kind of paraphrasing it as discovered. So somewhere something is there waiting to be discovered, because I think that we all are growing in a world of dogmas. Even when you read a textbook, it's all based on dogmas. Many of them are proven theories, but they're still very, very dogmatic in a statement. And many of them are not really 100% proven. So, for example, when I was growing up in science, we grew up with this dogma that there is no lymphatic drainage from the brain. So the sewage pipes taking waste products from every tissue into the lymphatic circulation to kind of detoxify the tissues. The belief was that there is no such vessels around the brain. And of course, 10 years ago, we were lucky enough to find these vessels and to prove this dogma incorrect. And I always quote a paper in a medical journal called Lancet from 2003, where they say that a lack of lymphatic circulation in the brain was described in details in whatever year. And I always ask myself, how do you describe a lack of something in details? And how many other things are out there described to not exist? And they're just laying down there and waiting for somebody to look under the rock, and you find a whole new world. So it's quite exciting.
Absolutely. I love that story. And I think you touched a little bit on your background and kind of how you got into science. I know your interest began in cancer research. Can you talk a little bit about what sparked your interest originally in this area of medicine or in science at all?
Well, it's funny because I grew up in a family of physicians. So my father is a doctor, is an MD, and my maternal grandmother, who was a very strong matriarchal figure in our family, she is also MD, and she was like our family doctor. She was a cardiologist, but we would go to her with anything. From my parents and their friends all the way to my children and their friends. And we would go to, she's still alive, and well, she'll be 100 years old next year, this coming summer.
That's wonderful.
We no longer use her advice in medicine, but anyway, so the idea of me not being a doctor wasn't really an option. So the family decided that I will be an MD, of course. But I remember myself as a little kid playing being a doctor, but I always played as the one who discovers medicines and not necessarily prescribes them. So this was a very kind of vivid childhood memory of mine. And cancer, I think even though I don't like those cliche statements, but I guess cancer probably was because my grandfather died from cancer. Both grandfathers died within the same year, 3 months apart at a very young age. They were in their mid-50s. One died from aortic aneurysm, the other one from cancer. So one was very, very fast, and one was very, very slow and very, very painful for him and for the whole family. So I guess his sickness probably influenced my idea of cancer, because neither my dad nor my grandmother were in an oncology field. So if I have to guess, I would guess that it was him who inspired me to think of cancer as a problem and to be willing to fix it. He passed away in early 80s. And so back then, we didn't even know whether cancer is infectious or is it not. No, and there was all kind of weird theories about it. So I do remember that. I remember that I was growing up and I was worried that somebody will cure cancer before I had a chance to.
Well, there's still time, right?
Yeah. So then when I went to Weizmann, I joined a very, very strong lab that was working on cancer. Moshe Oren, he actually cloned one of the first major tumor suppressor genes, p53. And I thought I would actually stay there. But then, like I said, I heard this presentation about immune and brain, and I just fell in love. So I never went back to cancer. Now we're kind of looking a little bit into brain tumors, and I am really fascinated by the topic.
Very interesting. And then how did you make the decision to come over to the U.S. to continue your training or take this faculty position at the University of Virginia?
So in Israel, it's not even an option of not going abroad to Europe or to US. Of course, US is preferred. So I knew that I was going out for at least 4 or 5 years. And I came and I loved it. I loved the freedom of doing science. I mean, these are small things, but then I think if you have listeners from outside of US, everybody will understand that you order a small reagent, and then if you're lucky, it comes in a week or two. If you're unlucky, it comes in 3-4 weeks. And here, if you are unlucky, it comes in 2 days. And normally just go to the next building and you go into this, what we call mall mart for molecular mart, a room, and you just pick up your reagent of interest. You order it online and it comes next day. So the way of doing science in the US, it's easy, it's comfortable. I've had a second lab in Germany for 5 years before COVID. It was a very small lab, very small operation, but I was still involved, of course, with ordering materials and reagents and all this. And I had trainees there, and it was uncomparable to how we do things in the US. I mean, it was brilliant science, brilliant colleagues, wonderful trainees. They think wonderfully, but just the pace is very, very different. Again, because of flexibility of reagents and of machines and devices and whatnot. So I'm a big patriot of United States and also of US science. And I just hope that we will maintain that leadership in science in the US. And that, of course, to maintain leadership in science, we need infusion of funds, which we are benefiting from right now from NIH as a major source. And I just hope that our leaders, whoever they are in the future, understand the importance of that support. And that's what allows us to lead the science in the world. I mean, there is no argument about that. Today, I think the leaders, I don't want to say that best science is done in the US, but I think that most of best science is done in the US. We are absolutely leaders of science.
Yeah, I think having that support is critical. And what then ultimately brought you mentioned you were at University of Virginia for a number of years. How did you end up here at Washington University in St. Louis?
I was a chair in Virginia, chair of the department. I had a wonderful department, really exceptional colleagues. And I loved Virginia, and I loved UVA, and I loved Charlottesville. But maybe two main reasons. One is a little selfish. I realized that I was in my maybe prime time in my 40s, and I probably have maybe, I don't know, good 10, 15 years to really do some exceptional science. And I wanted to maybe withdraw myself from administrative work. And I think it's always good for ex-chair to move away somewhere and not to stay and bother whoever the next chair comes in basic departments or clinical departments on medical campuses in the US, because I think it's a little different elsewhere. But maybe the main reason why WashU is because I think WashU provides an absolutely unique opportunity to collaborate with clinicians. As a joke, I always say that every clinician at WashU wants to be a scientist and every scientist wants to be a clinician. And I think it's very true. And also, whatever crazy idea we have or whatever sample or whatever idea to discuss with clinicians, let me shut it down and tell me this is like the weirdest, the craziest thing they've heard, and then there's no way to go, but they will listen. And we need to get the particular tissues and preserved in particular way. I mean, they're very, very open to interact with us. We hopefully, knock on wood, may be getting into clinical trial at some point soon. And again, we think that WashU will be a fantastic site for that just because of how open they are. And I think WashU is amazing on many aspects, but I think that for neurodegeneration and for immunology, this is really probably one of the very best programs in country. So for me, coming in a place that has very, very strong immunology and extremely strong neurodegenerative disorders kind of aspect of neurology, and then combine the two areas and develop something new and exciting like the center that we have was really a very exciting opportunity. And then I remember I was talking with some of the leaders here, again, without naming names, or good friends also now. And I told them, what if I regret? What if I move? And then 2-3 years down the road, I will say, no, I really regret coming in. And he told me people more regret for the things they haven't done than for the things they've done. And I think it was another inspiring moment for me. And so no regrets. I think that some of the works that we've published since moving to WashU, we would have been unfortunately not able to do it at University of Virginia or many other places, simply because of that interaction. It's very, very close interaction with clinical people.
That makes sense. And perhaps we can talk more about your work next. Is there a particular project that you are working on at the moment, Jony, that you are the most excited about and want to share with us today?
So I have 3 kids, and each one thinks that I like the other 2 more. Mm-hmm. Same in the lab. I have a bunch of projects, and everyone thinks that I like the other projects more than I like their project. So I will not be telling you which project is my most exciting one, but I will tell you that we just published recently a paper, maybe because it's a most recent paper I can discuss. Actually, it came out today in print. It was published a few weeks ago online in journal Nature. And this is kind of our first attempt to use cellular immunotherapy for neurodegenerative diseases. Of course, we know about CAR T cells that are used for cancers, and their concept is a little bit simpler because we have T cells that can attack and kill cancer. So we can take those cells and trick them so that they will start recognizing cancer cells even better, and then they will go and will attack the cells and kill them. So when we have a condition of neurodegeneration, or in our case, we just did it on CNS trauma, on spinal cord injury, you don't want to kill beyond what's already been killed. So instead, you want to go there and make the environment as fertile as possible so the damage doesn't continue to grow. So remember, CNS injuries in the brain and spinal cord are very unique because every time there is a damage, of course, what's being damaged will inevitably die. But areas around damage, which have not been damaged by the initial trauma, will continue and also degenerate. So that's a problem with stroke, problem with injury. It's a problem with every neurodegenerative condition. So we have a spread of damage, or what we call secondary degeneration. And it's been shown by people that if you work on the environment and you make environment a little bit more supportive, for example, you allow blood vessel regrowth, you allow this cleaning crew to come and clean the area more efficiently, then you can prevent the spread of damage. So we had a goal in mind to take these immune cells and instead of making them kill something, go there and organize the environment and make the environment more permissive for neuronal survival. And we were able to do it, and we showed this in mice, and the results were quite amazing. Basically, we believe that the immune response to any problem is, at least initially, it is aimed to be beneficial and protective because the immune system is our maintenance system. Of course, things can go out of control and the immune system may become detrimental, in which case you want to shut it down. But the initial immune reaction is most probably beneficial in all conditions. So with that in mind, we took those immune cells out after very few hours, days after injury. We analyzed what these cells look like, and we said, what if we just now multiply those cells, make a lot more of them, and inject them into injured mice? Will that be able to help us? And long story, the answer is yes. Of course, we did some modification to them to make sure that they will not turn into detrimental cells. So that was working really well. Basically, mice which were paralyzed were now recovering, walking, not perfect walking, but they were much, much more improved. And this is exciting also because, again, in collaboration with our clinical colleagues from Neurosurgery department here at WashU, we were able to get cerebrospinal fluid samples from patients with spinal cord injury. Actually, we get samples every day after injury for the first week. And even though we don't have, of course, data to show that, yes, these cells are beneficial for these patients, but we do see that the phenotype of these cells is very similar to what we see in mice. And in mice, they are beneficial. So hopefully, one day soon, maybe we'll be able to go with the cells to again take it from individual after injury, soon after injury, expand them, and then deliver them back in the hope to ameliorate the damage. And we're now testing the cells in other diseases. And one that came immediately to mind was ALS, or Lou Gehrig disease, because it's also spinal neurons, or motor neurons are dying in spinal cord. And we see some really exciting results there as well. And this is just the first step in this immune therapy for CNS diseases. So I think that this is a very promising and very fertile ground, which will probably take off. And I know that there are already several companies who are looking into it, and we're trying to launch a company as well. So I really believe that this will be the future of neurological disorders. Again, it will not probably cure Alzheimer's or ALS, but if we can slow down disease progression, or even better, if we can delay the onset of the disease, I think that would be fantastic.
Well, Jony, I think this is really remarkable work. And of course, it has tremendous potential for impacts, like you said, in spinal cord injury and beyond, starting to look at these other diseases as well. And I know scientists doing important, high-impact research depend on their suppliers providing high-quality research materials and products. And we'd like to take a moment to thank our sponsor, Innovative Research, and to talk about their portfolio of human coagulation products. From Factor 12 to fibrinogen, Innovative Research provides scientists studying hemostasis and thrombosis with the products they need in the quantities they desire. Contact Innovative Research via email at sales@innov-research.com. That's sales@innov-research.com to find out how their team of experts can provide human coagulation products for your research study today. And returning to our interview with Jony, it's not always success in science, as you probably well know, Jony. And I think most of the time, the public doesn't get to see the struggles, the failures, and all of the things that lead up to these really exciting discoveries in science. So can you share with us a time where you struggled with something, or you perhaps had a major failure in science? And could you perhaps explain what happened and walk us through how you got through it?
So first of all, most experiments fail. And to be honest with you, if experiments don't fail, that makes me very, very suspicious. If somebody shows me results which are exactly what we're expecting, and it's like experiment after experiment, I usually ask somebody in the lab to go and do the experiment together with that person. It doesn't happen often, but when it does, I get very, very suspicious because most experiments in science are failing. That's why we have to reproduce everything. That's why we're doing it blind way. So, for example, when we treat our animals, we don't know which animal gets which treatment. Sometimes it's unavoidable because of some other genetic issues where you clearly see the diseased animal versus not. But when it's possible, and it's often possible, we try to do assessment as blind as possible. When it's not possible, we are asking two people to assess, one who is in charge of the experiment and the other one who is just a lab mate, just to make sure that it's as reproducible as you can. Again, like I said, because most experiments are failing. With the failure, you need to be also very careful because let's say you have a hypothesis and you are testing it and the results are negative. Is it negative because the experiment did not work technically, or is it negative because your hypothesis is incorrect? So I tell always people that your negative result, you also need to do that. So it's not like if it's negative, it's absolutely right, but if it's positive, let's reproduce. No, if it's negative, let's reproduce it as well. Just to make sure that we are always continuing on the right path. And I always tell students when they come to me and say, oh my God, my experiment failed. I have people like bursting in tears in my office because experiments didn't work and because their hypotheses were all slain. Like I was saying, beautiful hypothesis slain by ugly data. I always tell them that if all experiments work, then PhD would last for maybe 6 months. And PhD goes for probably at least 5 years, sometimes it's 4, and often it's 6 or even 7. But if everything worked, then we would be done in 4 months. I would write the paper, submit, and be done with this. And probably I would say that most papers at the end, that same first author can reproduce everything in probably less than 6 months. But the time it takes to get to the experiments to working repeatedly, understanding precise conditions and all, that requires a lot of patience and perseverance. That's why I'm saying something which is not very popular is science is not for everybody because you're dealing with constant failures. And remember, we are also today, for example, in science, the kids who come to science are usually brilliant kids, most of them, maybe all of them. They never failed until the time they got into a lab and in their experiments. So very often we have these brilliant kids who just don't know how to deal with failure in life because nobody teaches them. They all get, through high school and through undergraduate with 4 O's, and they believe that everything they touch is bound for success. And it's not. These are all real struggles. Sometimes you would feel like, oh my God, my world is collapsing on me. I had a student who in his 5th year had to close his project. Funny enough, we now understand that his project wasn't incorrect because there is another lab, actually 2 labs, that eventually showed results which were very similar to what he was showing. So he wasn't wrong, but at some point experiments stopped reproducing and nothing worked. And he just came to me, big guy who used to play football in college and was crying in the office, and I was crying with him because I felt the pain he was experiencing. But on the other hand, what can you do, right? So things sometimes don't work, and that's part of the world, part of the life in science. Of course, you want to catch them early enough, and if the project is not going, then don't push it, right? That's why I always tell people, at the beginning of the project, the initial experiments that are laying the foundation, if they are shaky, if they are not hardcore reproducible, then drop the project. You don't want to build your castle on a foundation which is all rotten. And this is super important. I myself experienced, a bunch of failures as a bench scientist, as a student. Yes, I would always say that I need to have 5 projects, so one of them at least succeeds so I can publish something. Of course, science back then, 20, 25 years ago, was a little bit different because you could have had one tiny small discovery, you beautify it a little bit with some results around it, and then you submit your story and you publish your paper. Today, to publish a substantial paper, a lot more work needs to be done. I think the criteria for acceptance, in my opinion, today are much stricter than it was before. You just need to provide a lot more evidence course, we have a lot more scientific tools are much more advanced, so we can provide this evidence that we maybe couldn't 20 years ago, but requires a lot more work. So the projects are much longer. And then at some point when you realize everything I was doing is actually not what I was thinking, results are the results, right? So if your result is not reproducible, that's a big problem. If you're faking your results, that's a whole different level. And I really hope that we don't have many of these people in science. And unfortunately we do, but not so many. But interpretation could change. And I think you need to be open. I think one of the worst problems is that people are married to their hypothesis. They have theory and they married to it, and they try to make results support the theory. And I always say that results are unbendable, but theories are, and I can bend my theory to whatever results I have. So I think once people realize that, you will get unburdened, not to quote anybody, but you do get unburdened by this idea that everything has to fit into that hypothesis. Unfortunately, some principal investigators really want everything to work the way they thought about. I mean, that's a problem. That's not how science should be done. My favorite results are those that are showing significant difference between two groups, which is opposite to what you expected initially. When I see results which is different to what I thought, we just got this. We just got the group, and I was like so happy. I told my postdoc what an amazing result and all this. And I said, it's amazing, really a brilliant idea, brilliant story. He came to me for one-on-one and he says, I don't understand why you're so happy about it. I said, what do you mean? Look at this difference. And he said, yes, but did you read the groups? It's reverse. And I said, oh, it is reverse. But it's even more exciting now because it's definitely real. Let's just make sure that it's reverse, and that's fine.
Let's think about it.
Yeah, exactly. Yeah. So you constantly deal with those failures and people who can't. I mean, it's a problem. And interestingly enough, even though I had in my lab absolute brilliant people who are amazing scientists, and one of them just now got his own independent lab. He was very young when he came to my lab. Usually I do see that there is some sort of advantage when people come a little bit older, a little bit more experienced. So if people are listening, I would very much advise undergraduates, don't rush into graduate school. Take a year or 2 or 3. Do your postbac research, get some experience your failures. It's better to fail on somebody else's project than on your own project, right? Learn how to deal with failures and then apply. Yes, you are, quote unquote, wasting 2-3 years. I think you will gain those 2-3 years and probably more during your PhD and then postdoctoral studies, because you just come more prepared. And also ask themselves very honestly, do I enjoy it? When I go to bed, do I think about this project? I mean, is this really exciting? Because I honestly think that without being obsessed about the science and about the project you are doing, I think it's very hard to continue in science because of what you asked, because of all these failures. So you really need to love it to deal with all the failures around you. And again, not everybody is in it. And I see that people who are very successful in my lab and in other labs, these are people who constantly think and talk about science. They're living their science. And I think that helps tremendously to deal with failures. If you have a problem in your family, you keep going, right? That's your family. So if you have a problem in your science and you truly love your science, then you keep going. You keep fixing the problem and trying to deal with them as they come.
Well, I think you brought up some really great points. And I think just hearing you share your experiences helps normalize failure, but we don't want to dwell on just the failures. I'd love to talk about the successes as well. So, Jony, do you have a favorite success story in science, whether it was big or small?
There are a bunch of success stories, and thankfully, luckily, that we've experienced in maybe the last 10, 15 years or so. Well, there are several. Maybe the funniest one, it was from my graduate work. I think I just finished my graduate work and I stayed in the same lab for postdoc. And so I had this crazy idea that mice without functional immune system, I thought, would not be able to deal with stress, which was showed. And then I thought, okay, what if immune system supports a bunch of other organs? So it probably supports also the brain. And if it supports the brain, in mice without functional immune system, the brain will not be functioning as well. So the learning and memory will be impaired. I mean, crazy idea, absolutely insane. But I was lucky to work in a lab with Michal Schwartz where crazy ideas were more than welcome. And I tried to mimic that, and crazy ideas are more than welcome in my lab as well. So we actually could not do behavioral experiments in Michal's lab because we were more like a cellular, molecular, neurobiology lab. So we sent our animals to a group of scientists in another institution in Tel Aviv University. We sent 2 groups of mice which were normal mice and mice without functional immune systems. And we didn't tell her what the experiment was about. We just told her we have two groups of mice and we think that the one with maybe some issues with one mouse, with one group. We did it because we wanted her to be as neutral, as unbiased as possible. And so she called us and said, I don't know, one group of mice are real idiots. And so we opened the codes and the group without functional T cells, T lymphocytes, were the idiots. So it was so exciting, but that's not enough. So then we took two groups of idiot mice, so to speak, two groups of mice with immune deficiency. And one of them, we gave back immune cells, which we thought were missing, but to mediate this effect, the T cells. And we sent her two groups again. So now one group is the background of a stupid mouse, and one group is a stupid mouse plus the immune system, which they didn't have, which of course we were hoping to get some improvement. And she calls me and said, experiment didn't work. And I said, oh, wow, what happened? She goes, I still have one smart group and one stupid group. Mm-hmm. And I said, okay, that's real. It's working. I remember the moment. And then we sent the paper to some of the top-notch journals, and they rejected it because they said there is not enough novelty or whatever they said. Anyway, that was really amazing feeling. And the second one was in 2014 when a postdoc of mine discovered lymphatic vessels. And he called me to microscope, and because he was doing a perfusion of a mouse and then was looking in the borders of the brain by whole mounts. And we saw some vessels which contained immune cells, which didn't make sense because mouse was perfused. So all the blood cells should have been out of the blood vessels. And we see these two vessels that are filled with immune cells. And I mean, it was like weird. So we said, okay, this could be lymphatics, even though we know that they shouldn't be there because that's what the field states. And that's what all the textbooks state. So we went to colleague, asked for markers to label for lymphatic vessels, and just to kind of prove ourselves that we are wrong. And so he put these markers, and then he calls me to microscope and he says, come see, there are lymphatics there. And I do remember that moment. That was incredible. I remember myself now coming out of that room and saying, okay, this is real eureka. This is something that I wouldn't dare to even dream to experience in my life. So this was a very, very fun moment as well.
Wow. I think those moments where, like you said, the results are just so clear are few and far between in science, but you have to take a moment to celebrate them.
Yes, exactly. Which we did.
Oh, good, good. And I think having these exciting projects and these exciting results is one of the great things about careers in science, but the travel is another thing that really is a highlight in the career that many people may not realize is part of it. So do you have a favorite place, Jony, that your science has taken you?
Yeah, we travel a lot. We travel a little bit too much. Every travel, you learn so much, right? Whether you're going to visit university and then you meet with colleagues or graduate students and postdocs for lunch. And then usually I come back with amazing ideas and some new knowledge. Or you're going to conferences, and if it's a good conference where unpublished data is presented, then again, you learn a lot. It's like reading nonstop for 5 days, but instead they're just telling you the stories about their work and you're hearing it from the first source. It's incredible. But I'm lucky. I've been to a bunch of places. So I was born in the Republic of Georgia, which was back then a Soviet Republic of Georgia. And in 1990, we left the country and moved to Israel. And since 1990, I haven't been back to Georgia. And then in 2017, there was a meeting there and I was invited to speak. And I guess that maybe that visit was very special just because, 27 years coming back to the country where you're born, it was very, very special experience. But I love traveling to the East. I mean, China and Korea, the culture is so welcoming and so exceptional. And I love going to those countries. I was missing it quite a lot during COVID, but now, thank God, we're back. I don't know. I think I have like one place where I love to go. Actually, next week, I'm going to see some amazing lake, apparently, in the Alps in Germany. So we're looking forward to it. Yeah, there's lots of interesting places where scientific meetings take place, and I think it is very important because some of these small meetings in really exciting locations, you just get together with a group of people who are like-minded or not, but they are interested in similar topics, and you just continuously discuss. And if people are open for exchange of ideas and maybe even arguing, agreeing, disagreeing, I think it generates a lot of new ideas, creates lots of new projects in the lab. So I think scientific travel is extremely important. And if anybody thought that Zoom can replace it, then they were delusional. So today, if I'm invited to give a talk by Zoom, I decline.
Well, I think that's remarkable. You've been able to travel to quite a few places. I love this story that you shared of being able to finally go back to your birth country in Georgia. Was it exciting to sort of see places that you perhaps remembered?
Yeah, I did some, and we traveled. So I also remember the places where we would go for our summer house and all this. So I took a guide and we traveled. And then in 2019, I came there with colleagues. I was co-organizer of a meeting in 2019. brought a bunch of colleagues. We traveled again. I've been going since then almost every year. And we just came back. We were there in June again with the conference. So trying to bring American science to Georgia. Unfortunately, no, it's still a developing, maybe redeveloping country. I think science-wise, unfortunately, it's not as strong as it used to be. But I think they are making efforts, and it's really exciting to see these conferences going there. And a bunch of people came from different countries, from Europe, from US, from Asia. And so we had actually a very exciting 3 or 4 day meeting. It was the Immunology Center. Then we had our Neuroimmunology session. It was wonderful.
That sounds wonderful. I think the people in science are another thing that makes these trips a lot of fun, being able to meet scientists from all over the world and just even in your own institution, the diverse group of people that you get to work with over the years. And I think so many cases, these scientists that you work with do not meet the stereotypes that are out there in the media, where it's always somebody in a stiff lab coat being very serious and working all the time. I think some of the most funny, creative, amazing people that I know are scientists. And I think these quirky traditions spring up in labs, and you just share some funny memories together over the years. So do you have an example, Jony, of this human side of science, whether it is a tradition or just a funny memory that you can share with us?
I think we were last week or 2 weeks ago, we were at this huge conference for neuroscientists called Society for Neuroscience. And after our social, we went for a dinner with a couple of colleagues. I mean, we were sitting there maybe for 3 hours. We did not stop laughing. My facial muscles were in pain next day because we were just laughing like crazy. And we were not drunk or anything. We were just laughing because it was just good discussion. But actually, 2 weeks before then, it was in September, actually not so long ago, it was the 25th, 27th of September. We had a conference here in St. Louis, and we called it Rethink Neuroimmunology, and it was our 3rd year. So we were in St. Louis in 2022. We went to Weizmann in 23, and then we came back to St. Louis in 24, and we're going next year to Boston and then hopefully France, and we'll see where we're going next. It's a tight group. I mean, we bring new speakers all the time, but it's a tight group of core investigators. And St. Louis, we have this very quirky, it's called the City Museum. Just Google it up. It's really funny. It's like a playground for adults. So all the serious scientists, after a whole day of discussions and after dinner, so we all ate and in a really fancy place, we're all coming there. Nobody knows what to expect. And I tell them all, guys, you absolutely should come even for half an hour. And then, so they left with buses, and I was kind of coming at the end just to make sure that everybody was already there. So I come at the end, and as you go, there is this huge slide that kind of brings people from somewhere, and I don't know, whatever floor, all the way down. And I come and I see one very respected scientist stands there barefoot, and she's pulling out by the leg another scientist because he was a little stuck in that slide, And then from there, it just went funnier and funnier. And they put me to go through some tunnel, and then we're taking pictures and we're making some crazy comments. Some of them are online. So I was in this tube, crawling through the tube. So somebody made a comment, said, Agent 007 crawling through lymphatic vessels and things like that. Scientists, like you said, many of them are quite funny, and they could be very serious and very much into our problems. But then usually when they get together, and very often it's with trainees and with postdocs and students and whatnot, right? I mean, we let it go a little bit, and then it's funny, it's entertaining, and it's also a little quirky as well.
Definitely. I think the City Museum is amazing. I've been there myself.
So you know what I'm talking about. Yeah.
Yeah. I think it really highlights the sense of humor and that sense of camaraderie, which I think is so important in science, because you are facing some tough challenges, solving some difficult problems in the lab every day. And I know oftentimes, Jony, unfortunately, things like funding, staff, technology, feasibility, et cetera, hold you back. But if we took away the barriers, what is the one research question that you would want to answer most?
I still think that, again, brain is defined as immune privilege, and we're not sure understand what it means, but if you ask 10 people, they will give you 11 different answers. But the idea is that the brain has unique status with the immune system. And so even though now we know they do interact, but they interact to a particular degree, not like every other organ. And the mechanisms to explain why this is so was with 2 levels of explanation. One level was the blood vessels within the brain have a unique barrier called blood-brain barrier, and immune cells cannot really easily cross that barrier or cannot cross the barrier at all, and you don't have immune cells in the brain. Well, that is not right. So this is not true, and there are many labs who showed that if you activate immune cells in a particular way and they need to get into the brain, they will definitely cross that barrier. So that's not explanation. And the other explanation was that we do not have lymphatic vessels anywhere in the brain, and therefore there is no direct physical communication between the brain and the immune system. That is also incorrect. As I told you, we discovered lymphatic vessels around the brain that serve the brain about 10 years ago. So now two major underlying mechanisms to the question of why brain is immunologically unique are not correct. So what is it that allows us a very, very unique interaction between the brain and the immune system? Why cancers in the brain are not rejected if we do have lymphatic drainage and if immune cells can cross the barriers? So why don't cancers in the brain get rejected as efficiently as they are in the periphery? Why CAR T cells don't work against brain tumors, right? So why even checkpoint inhibitor therapies are not working for brain tumors? So this is still a very big question. We actually have a paper coming probably next week or maybe in 2 weeks online that kind of gets a little bit at it, maybe provides a new level of mechanism. So basically, brain sends this unique self-derived molecules to present to the immune system. And when the immune system reacts to them, they generate immune cells which are more of suppressor than inflammatory cells. So normally immune responses are either effector cells, so they go after their target, or they are more of a regulatory cells, so they more suppress and regulate the immune response. And so these molecules, these brain-derived peptides, are inducing more of a regulatory immune response, which has never been shown before. So we call them guardian peptides. So these are self-derived proteins coming from the brain, and for whatever reasons which we're showing in this paper, are inducing this very different and very unique suppressing immune response. And of course, in disease conditions, they're disappearing, and potentially one way to help with the neuroinflammatory conditions is to restore those peptides. So this is one very important layer, but I don't think it explains the whole thing. So if I could have one answer, I would love to understand that. And of course, the question is, how can we efficiently treat neurological disorders? I believe in immune therapy for brain disorders, but what should be our target cell? Again, we're focusing on particular cell types, on T cells, but is this the right target cell? Well, it's a target cell. Is this the target cell? If I had a magic wand and I had one question, I would, of course, ask what are the mechanisms for CNS immune tolerance or immune privilege. If I had two, I would ask, okay, which cells I should going after to develop the best, the most successful, the most promising immune therapy for brain disorders.
Well, Jony, I think this is all fascinating, and listeners will have to keep an eye out for this upcoming paper. And we've talked about a lot of different things today. I'd love to wrap up by just touching on advice. So for listeners out there who might be on their own scientific journeys, what advice would you give them that perhaps helped you along your own journey?
So the advice I give everybody is to not listen to anybody's advices. And I think it's a very important thing to realize, because there are people who love to give advice, and often they confuse advice with order. And you give you advice, and then if you don't follow, they get upset with you. Why did you follow my advice? We also need to remember, right? So sometimes we're looking at people who are saying, oh my God, he or she are so successful. I want to be like her or like him. Let me ask them, what are they doing? So people go and say, so what do you do? How do you succeed? First of all, I think success is a very weird term. What's successful? I don't know. I think you could be successful today because you got some really amazing results, and tomorrow you're not successful because the data is not producing, for example, right? So success is an arbitrary unit, in my opinion. But let's say you say, okay, I would love to be like that person. And so you go and you ask them for advice. They tell you whatever they do. For example, I have the luxury during the day, I can spend time and meet my people in the lab with postdocs, with graduate students, discuss with them ideas, go to all kinds of meetings. And then at night, I am very functional. And so I can actually do a lot of work done during nighttime. But if I give this advice to somebody who absolutely needs 8 or 9 hours of sleep and who absolutely cannot function in the evening, and then try to follow my advice, they will collapse within a couple of weeks. So I think everybody has intuition. You just need to listen to your inner voice. And if the universe tells you to not do something or to not go that way, and that maybe it's not for you, I think you should listen to it. If all the signs are saying no, then maybe, or saying yes, maybe you should listen. Just listen a little bit more to your inner voice. And I really believe that if you are not excited about the project that you are doing, about scientific question you are asking, I'm not saying it's impossible to do science and to do it well. I just think it will be very, very difficult because of all the failures that you will encounter that we discussed before. So listen to your inner voice. Don't listen to many other advices, and just try to honestly ask yourself, is this the right path for you? Is this my calling?
I love it. I think that's a wonderful message for our listeners. Is there any other last note of inspiration you'd like to leave everybody with at the end of our conversation today?
No, just please read this book. And the imperfection in science and even in life, I think it's okay. It's fine to be a little bit imperfect. Sir Peter Medawar, who is Nobel laureate as well, who also wrote a bunch of books. And another books, I don't know if that was mentioned to you, is called Advice to a Young Scientist by Peter Medawar. I guess that probably was mentioned before. So there he says that it's very important to plan experiment and plan it well and think. But at the end of the day, at some point you need to get up, go do the experiment, fail, and then plan better, and then fail better, and then do it again. You can't just sit there and plan experiments forever. It's okay to do things, and it's okay to make mistakes. And if you work in an environment that is not mistake permissive, then I think it's a wrong environment for you. Mistakes should be allowed. Everyone makes mistakes. Even DNA polymerase makes mistakes sometimes.
Well, Jony, that is a wonderful note to end on. If our listeners would like to learn more about you and more about the work that you do, what is the best way for them to do so?
Well, unfortunately, there is a lot of information about me on Twitter and on some other social media, but there is a lot of publications for people who read scientific literature. There is also, I think, a published Scientific American a couple of years ago. People can read that. So I think there is enough material, unfortunately, online about the lab and what we do in the lab and what ex-lab trainees are now doing in their own labs, which is also another source of inspiration for me, people who were trained with me and are now independent and doing some really exceptional science.
Perfect. Well, listeners, definitely check out these resources. And Jony, thank you so much for joining us on the show. It was such a pleasure to chat with you today.
Thank you very much for having me.
My pleasure, Jony. And listeners, thank you so much for spending time with us today. We hope you join us again for the next episode of People Behind the Science.